called fast switch or type 2. And actually, there are a variety of subtypes of the white fibers. There's a type 2A, type 2B, and type 2X. You remember that? Okay. They have some slight differences in what they do, but basically they're larger in diameter than reds. They have fewer mitochondria. They have a lower concentration of myoglobin. They don't really display or show, if you use the same substrates, the same kind of reactivity for the substrate. They don't produce as much of a colored product that you could identify. So these are called fast switch or type 2. They fatigue rapidly, but they're adapted for a very quick and powerful contraction. So I suppose if I had a career that was destined to be a power lifter, I would like to have a larger percentage of white fibers in the muscles of my upper limb, maybe my lower limb as well. And before I go any further, let me just say that in most humans, we have about a 50-50 mixture of both in almost any muscle you look at in the body. The rare exceptions are gifted athletes, Olympic type athletes, our graduate students, usually have, for whatever their sport, they seem to be adapted and have a preponderance of that fiber type that would help them in that sport. Can you change fiber types? And the answer probably is I don't think so, because it appears that the fiber typing is set by the nerves that innervate them. So you'd have to rearrange or reconnect their axons, and I'm not sure that's doable. It occasionally happens when you do a myocutaneous flap. Usually a myocutaneous flap is something you take from one area of the body to cover another area where there's been connective tissue, skin, and muscle loss. You almost always save the blood vessels and nerves that come from that area of donation so that they can be reconnected in the new area. In some cases, those reconnections involve a transposition of axonal fibers or nerves, but that's pretty rare, and you're not going to do that with an athletic intention involved. So it appears they're probably not modifiable. You can change the metabolic activities of some of the fiber types through exercise, but when you stop the exercise, I think they go back to the original fiber typing that was there. Yeah, there's a lot of experiments regarding that sort of thing. Let's now talk about how myofibrils and myofilaments are organized in a muscle fiber. So and I'll show you an image of this breakdown in a second. If you take a muscle and just cut it out of the body, I'll donate some of my deltoid. As Brian refused. If you look at that on a low power scope, what you're looking at are muscle fibers. So muscle fibers comprise a large muscle, a named muscle. If you take each muscle fiber, it's composed of a large number of smaller subunits. Again, they're sort of round, they're called myofibrils. And then if you look at a myofibril, again, you probably need an electron microscopic image to do that. They're composed of two populations of contractile myofilaments. These are myosin and actin. The thicker ones are called myosin, the thinner ones are called actin. I'm sure you've heard this 100 times. Because the thick and thin filaments are registered and always maintain pretty much the same position in a contractile subunit that I'll talk about in a second, that gives you, in a larger view, a less high power view, the characteristic banding pattern, the striated banding pattern of skeletal muscle. Here we go again. Come on. Okay, so here's how we can break that down. So here's my upper limb. I'm taking a hunk of my deltoid out. If you just look at it, almost even under a dissecting microscope, you can see it's composed of muscle fibers. Take an individual muscle fiber and cross-section it. You can see it's composed of a number of smaller subunits, narrow in diameter. Those are called myofibrils. So, am I the only one that's just that moving? Okay, so this thing It's kind of pinkish purple in the center. That's a myofibril. You'll notice a couple things about myofibrils. One, they show this characteristic banding pattern that's got A bands, I bands, and a Z band or a Z line. And there's a term, because if you look at enough of these myofibrils, you'll realize that there's a unit that repeats over and over and over again in a myofibril. It's called a sarcomere. That's the repeating contractile unit within a myofibril. So a myofibril is made up of thousands, millions of these sarcomeres. Pictured here. What's a sarcomere? The sarcomere is then the distance from one Z line to the next Z line. Those represent the beginning and end of the sarcomere. The lines, the bands that you should be able to identify are a very light staining area called the I band, a more dense staining area called the A band, and in the center of the A band is a M line, is an H area and an M line. I'll show you a better picture in a second. And then in the center of the I band is the Z line or Z disc. Okay. So if we would take one of those sarcomeres, just take it out of the myofibril and examine it, that's what pictured at the bottom of this image. So again, we've got a sarcomere from Z line to Z line. The A band represents an overlap between the thicker myosin filaments and their sort of club-like heads and the thinner actin filaments that are anchored at the Z line or Z disc. Okay? So here's our sarcomere. We take a sarcomere out. Here's our sarcomere enlarged. So here's the I band and I band. Here's the A band from here to here. In the center of the A is the H, kind of this light area, and the center of the H is this thing called an M line or M band. All right, so in this area we only have Z line material, mainly actin. In this area we only have myosin, but the area where the two overlap are this part and this part of the A band. Okay. All right. If you could take a cross section through any of these areas, if you took a cross section through the I band, you'd see this hexagonal arrangement of the thin actin myofilaments. If you took a section through the H band, you would see a similar hexagonal arrangement of the thick myosin filaments. But if you would take a section through this part of the A band, you would see they are arranged around each other. The thin actin filaments are arranged in a hexagonal pattern, typically around the thicker myosin filaments. Okay, but it got that. All right, so sarcomere is the repeating unit, functional unit between two Z lines of a myofibril. Thick filaments mark the extent of the A band, thin filaments mark the extent of the I band, where they're the principal myofilament. And the thin filaments also extend to the A band. H bisects or splits the A, the M bisects the H. Again, I was kind of running out of terminology for the slide. Can you identify these? Yeah, you can. Here's an actual EM image that shows one sarcomere. So again, we've got I band here, Z line or Z disc material in the center of each I band. Here's the A band from here to here. In the center of the A is the H, in the center of the H is the M line. So in the A band here and here is where we have the overlap between the thick and the thin actin and myosin filaments. Okay, so you can't identify these. You might be asked to do it again in a couple weeks. All right. The way that it's believed skeletal muscle contracts, the mechanism of contraction is called the sliding filament mechanism. So the basis for contraction resides in the interaction of the thick and thin myofilaments, obviously. And this interaction enables the thin myofilaments in a sarcomere to be pulled to the center of the sarcomere. That's how sarcomeres contract. So the heads of the myosin molecules drop down, they contact the active site on the thin actin filaments and pull the actin filaments toward the center of a sarcomere. When that happens, two things are shortened. Both the I band and the H band shorten as the sarcomere shortens. Those are the only two bands that change their length. H band and the I band shorten when the sarcomere shortens. Okay, so what happens during contraction? These things that look like the heads of a golf club, those are the head portions of the thicker myosin filaments. They need a couple of things to be around so that they can sort of drop down, interact with the sites where they're supposed to meet, these thinner parts, these parts of the thin actin filaments, and then on both sides, pull the thin actin filaments toward the side of the sarcomere so that the H band and the I bands shorten during contraction. Okay, so what has to be around for contraction to occur? Oh, here's a picture that shows those things. On the top panel, that's a relaxed sarcomere, resting sarcomere, maybe it's not relaxed. And then at the bottom, this contracted sarcomere showing the shorter I and the H bands. Okay. In addition to actinomycin, there are at least two, probably a lot more than that now, proteins that are called regulatory proteins. They're troponin and tropomyosin. So the tropomyosin is sort of a rope-like molecule, and the troponin is actually a complex that contains an inhibitor subunit, a calcium binding subunit, and then a tropomyosin binding subunit. Okay, so how do these work? So for contraction to occur, you need ionized calcium and you need ATP. So in this top part of the panel, when there is no calcium or ATP around, or both of them are not present in sufficient quantities, there's a conformational change in the troponin molecule so that the inhibitory subunit sort of interferes with, and that's not exactly the truth, but that's close enough for government work, the heads of the myosin molecule, so they can't see their appropriate binding site on the actin because this inhibitory unit has now moved tropomyosin in a way that prevents these two from getting together. That's what happens when there's no calcium and or ATP around. If calcium and ATP are around, which is usually the case, the calcium binds to the calcium binding subunit of the troponin. This again undergoes a conformational change. The tropomyosin subunit binds and moves the tropomyosin sort of out of the way. So now the heads of the myosin molecule can interact with their appropriate sites on the actin and split ATP to produce a very short movement in the swing phase of this activity. Get contraction. Contraction occurs. Okay. So here's a summary of that. Calcium has to be available for interaction between the actin and myosin, and after the contraction, it has to be removed pretty quickly too, as you might suspect. So what accounts, or what structure allows for the rapid delivery and the rapid removal of calcium? The thing that does that is the thing that used to be the endoplasmic reticulum in most other cells. This system is now called the sarcoplasmic reticulum, and it's arranged mainly in a series of tubular networks between and around among the myofibrils. So here's what that looks like. So what do we got here? We've got a segment of 5 myofibrils, okay? And they've shown us on this one at least one complete sarcomere. Here's the I band, Z line, the A band with the H and the N right in the center. So here's where the actin and myosin are overlapping with each other. So here's a system of tubules. I'm talking about these things and this thing. Though that's called the sarcoplasmic reticulum, a series of tubules. In different animals, they have a slightly different arrangement, but mainly what you've got is this random series of tubules around and between all these myofibrils. And in 2 places in each sarcomere, at least in human skeletal muscle, they have a very specific form. They form these larger diameter tubes at the junction between the I band and the A band. So at that I junction, we've got these two larger diameter parts of the sarcoplasmic reticulum. They're called terminal cisternae. Terminal cisternae. So there's one terminal cisternae here, there's a second one here at the AI junction on this part of the sarcomere, and there's another pair down here at this AI junction on that same sarcomere. Okay? Okay, so that's what contains the ionized calcium. There's also another very narrow tubule that comes in as an invagination from the plasma membrane of the sarcolemma. This narrow darker blue tubule, that's called a transverse or a T-tubule. So that means that every AI junction, there are two triads. This grouping of three, two terminal cisternae and one T or transverse tubule, that grouping is called a triad. So there's one triad here, a second one here. Okay, so if you think about it, what happens when a neuron, a neuromuscular junction is fired? You get a wave of depolarization that's coming down the surface of the muscle, and this T-tubule is carrying it inward among and between all these myofibrils and a muscle fiber. Okay? That signal that's brought in then induces a change in the membrane of the terminal cisternae and floods the area among and between all these myofibrils with calcium. If ATP is there, then we get contraction. Does that make sense to everybody? So, the site where we talked about this, you got the neuromuscular junction, the site where the axon terminal meets the muscle. Motor end plate goes by a couple names. So that axon terminal lies in kind of a shallow groove or a gutter. It's called the primary neuromuscular cleft. There are some other secondary clefts that come off of that. And then usually the myelin cover has stopped in the axon, at the axon terminal. Okay, there's still a Schwann cell there, kind of covering thing, but there's no myelin sheath anymore. So what you have at a neuromuscular junction now, as part of the process, is here's our axon terminal. You recognize it because it has lots of mitochondria and synaptic vesicles in it, clear core synaptic vesicles that contain acetylcholine. So here's the cytoplasm of the covering Schwann cell, but it's not making myelin at this point. Okay, so this primary synaptic cleft is this thing, this kind of shallow gutter here, that allows the axon terminal to kind of sit in a bit of the muscle fiber, which is here. There are some secondary clefts that come off of it, and that's what these things are. And then there's some junctional, what are called junctional folds of the muscle that kind of create these secondary clefts. Surrounding all this stuff is this kind of pinkish material, that's basal laminar basal membrane material. It does not appear to interfere with the transmission of the impulse from the axon terminal to the muscle fiber. And it's believed that both these primary clefts and these secondary or junctional clefts probably increase surface area to allow more neurotransmitter to interact with that localized segment of the muscle fiber. Okay? So what leads to contraction of a muscle? There's first discharge of a motor neuron, and again, that motor neuron is probably either in the spinal cord, the anterior horn of the spinal cord, or gray matter of the brain stem. That discharge of the motor neuron is carried as an action potential down the axon. The axon is myelinated, so there's no leakage of charge. I'm sure Dr. Segal will talk to you about that. When it gets to an axon terminal, what was a membrane phenomenon, an action potential, is now changed to a chemical phenomenon because we get release of a neurotransmitter, crosses a synaptic cleft, the primary neuromuscular junction, primary synaptic cleft, and then we get depolarization of the muscle fiber. Again, now changed from a chemical to an electrical phenomenon that goes down the surface of each muscle fiber. That potential is carried inward along the T-tubules, between and among the myofibrils. That eventually causes release of calcium from the terminal cisternae. The calcium binds to the appropriate part of the troponin subunit, It permits then interaction of the myosin binding sites on the actin, and you get formation of crosslinks between actin and myosin, and then the sliding of the thin filaments on the thick and shortening of the sarcomere. So that's what's involved in contraction of a muscle fiber. What happens to stop the process, or what's the reverse? Basically, the calcium is pumped back into the terminal cisternae. It's removed. Then we get a conformational change in the troponin binding site. That means there's no more interaction between the actin and myosin, and so contraction stops. And both these events, release and removal of calcium, occur very, very quickly. Okay, any questions about that kind of stuff? All right, good. Well, I said you need both motor and sensory innervations of skeletal muscle to make it work. There are two kinds of sensory receptors in skeletal muscle. The first one's called a muscle spindle. This is basically a stretch receptor. You can think of it as a set of springs in a little receptacle, if you want to think of it that way. This muscle spindle consists of specialized muscle fibers. Again, these are striated muscle fibers, but they're much more narrow than a typical muscle fiber, and some nerve terminals. There's a capsule that surrounds all this stuff, and the two kinds of narrow skeletal muscle fibers in a muscle spindle are called intrafusal. A group are called nuclear bag, another group are called nuclear chain. I'll show you, here's why they're called, and they have those terminations, or terms. So here's the spindle-shaped thing, that's the muscle spindle. It consists of a group of intrafusal fibers. In some cases, the intrafusal fibers, these are skeletal muscle striated fibers, have a group of nuclei sort of as a collection or a bag, so these are called nuclear bag fibers. These that are kind of strung out with nuclei are called nuclear chain fibers. So these are contained within this spindle structure. The regular components, the regular muscle fibers around here, they're called extrafusal fibers. Okay, so we've got this enclosed thing. There's two layers of capsule here, an external, internal capsule. There's probably some fluid in here, and also coming in and leaving are these various components of small nerves. So why are there nerves intervening this thing? Well, obviously we need some sensory fibers coming from these spring-like structures that tell when the muscle has been stretched. So that's what these red fibers are. These are afferent fibers. When these springs get stretched, they fire off and tell the spinal cord, hey, the muscle's being stretched. But you'll also notice there's some yellow and green fibers. These are called gamma afferent fibers that are coming in from the spinal cord. So those basically set the tension in the spring, so these springs are adjustable. You can set the tension as to how much you want before they fire. So these are essentially adjustable springs. So that's what the afferent or motor fibers are for. Okay, so that's a muscle spindle. Yeah, how can you recognize them in a tissue section? The only way I can really conclusively say, oh, that's a muscle spindle, is if I see them in cross section like this. If you see them in longitudinal section, they're really hard to identify. So again, sometimes the capsule tells me, oh, this has got to be a muscle spindle. And I think some of your slides have muscle spindles on them. Okay. The other sensory receptor in skeletal muscle actually occurs at the muscle tendon junction called the Golgi tendon organ. These are again encapsulated structures. They're found at muscle tendon junctions, although they're very hard to see based on my experience. They respond only to tension on the tendon. So these are only afferent or sensory structures. I don't have a good picture of one. This is a drawing, and I like the drawing better because it shows you that here's the end of the muscle fiber up here at the 12 o'clock position. Here's the tendon down here. So this encapsulated structure has got a bunch of collagenous fibers in it, like these chain arrangements, and the afferent fibers are starting in that, in between the loops of the chain. So when the chain tightens, the afferent fibers are sending up impulses that go back to the spinal cord and say, hey, wait a second, we're being stretched. Stop that, okay? I can tell you that in my research for my PhD, I looked at, like I said, 80 different muscle tendon junctions bilaterally right and left sides of rats, particular group of muscles in the rat. I bet I only saw 3, 4, maybe 5 of these Golgi tendon organs. So I think they're fairly hard to see. Now, maybe I just didn't do those sections, but darn, they're hard to see. All right, so much for skeletal muscle. Let's make quick work of cardiac muscle. Again, this is another kind of striated muscle fiber. The differences are that cardiac muscle contains individual cells that begin in this intercalated discs. The intercalated discs have, as their components, multiple examples of these kinds of intercellular junctions, some of which we've talked about. So in an intercalated disc, you can see a couple of fascia adherens, a couple of gap junctions, or a couple of macula adherens. They are variable as to the number of each of these at each intercalated disc. So like each intercalated disc doesn't have 2 examples of each, they could have multiple examples of each of these. Cardiac muscle cells show branching, they split. They contain large numbers of mitochondria, and often the mitochondria in a cardiac myocyte occupies the full length of a sarcomere. You don't see that in striated muscle, skeletal muscle. In cardiac muscle, the terminal cisternae and T-tubule are present, but there's only one of each. And the terminal cisternae is kind of, it's present on one side of a transverse tubule, but not on both. So you only have dyads, you don't have triads in cardiac muscle, and they're found at a different location in the sarcomere. They're found at the Z lines, not at the A junction. Cardiac muscle cells have a single nucleus that's centrally located. At one time, they were thought to be postmitotic. That means that they were thought to be incapable of mitotic division. That is wrong, and I have seen it myself in autopsy studies that I've done, where, for instance, a person has had a myocardial event that involved death of cardiac muscle cells and survived, lived for another couple of years or months, and then died subsequently from another event. Someone, got hit by a bus or something else happened. In looking at those hearts, I have seen the nuclei of cardiac muscle cells in mitotic figures. So I know they can undergo mitosis. The problem is, if you look in the literature, it's pretty well documented now, the number of cardiac muscle cells that can do that are very small, less than 1%. So there's something that will cause them to undergo mitotic proliferation, but just at a really very low rate. So if in your research careers, you guys find whatever those chemical substances are that cause cardiac muscle cells to undergo mitotic replacement, you should immediately notify Brian and myself, and we'll get on board with purchasing the stocks from that company before we go public. So they're not post-mitotic, but they're very low rates of mitotically capable cells. Cardiac muscle cells are not stimulated per se by nerves. There are no nerves in the heart. There are groups of fibers that cause the cardiac muscle cells to contract. They act like nerves, but they're really modified cardiac muscle cells. They're called Purkinje cells or P cells, 2 groups. And there are no satellite cells or equivalent stem cells in cardiac muscle. Therefore, there is, in reality, no potential for a regeneration of heart tissue, unfortunately. So if in your medical careers you deal with patients who have had myocardial events, where there's loss or death of cardiac muscle, they're almost always put on a cardiac rehab program, diet and exercise, because their hearts are further weaker. There's no replacement or regeneration of cardiac muscle tissue, at least to this point in time. Yeah. Here are 2 similar views that I showed you of skeletal muscle for cardiac muscle. The one on the left, a longitudinal section. The arrows are pointing to intercalated discs. You can see them at the light microscopic level. Sometimes cardiac muscle cells are binucleated. That's okay. That doesn't mean anything. These are cells that do branch and split. That means in a cross section, sometimes you get these really weird little profiles over here where they're very angulated or tapered. They're probably near branch points for that reason. They're not simply ovals or trapezoidal in shape. And again, you're not gonna catch the nucleus of all these, just do the vagaries of the section. Here's kind of a schematic of what goes on in a intercalated disc. Typically what you see are you get either immaculate adherens, desmosomes, or fascia adherens, much longer, zonular adherens if you want to use that term, more sheet-like junctions. And then in between the sides of cardiac muscle fibers, you get gap junctions. So what appears to be the case is that you get these adherent junctions, like fascia adherens or zonular adherens, near desmosomes. But the gap junctions almost always appear, in my experience, looking at cardiac tissue, at least under the electron microscope, on the sides between cardiac muscle cells, not at the ends. And it probably is because they're not exposed to tension during contraction of heart muscle. And this is the way that impulses spread from muscle cell to muscle cell through gap junctions. So it makes more sense put them on the sides rather than at the ends where they might be disrupted electrically. At least that's what I think. Here's an actual electron micrograph similar to what you got in your EM collections, showing macular adherens, fascia adherens, FA, fascia adherens, same as the zonular adherens, and then gap junctions mainly between the sides of these cardiac muscle cells. So if I showed you this image, you know it's cardiac muscle because of the gap junction. The mitochondria extend the full length of a sarcomere. That would help you. If I ask you where in this kind of tissue you would find terminal cisternae or transverse tubules, I hope you would say at the Z line rather than at the AI junction. So that's how I might ask that kind of a question. A similar schematic to the one I showed you for skeletal muscle. Again, we've got an intact sarcomere on the left side here, I band, Z line to Z line, sarcomere, Z line to Z line. Here's the A band, H and M. Notice the mitochondria showing the full length of the sarcomere. That's different than typically in skeletal muscle. Here are the dyads. So here's one transverse or T-tubule. You tend to have in cardiac muscle a terminal cisternae on one side but not both. So on one side but not both. So at Z lines you get dyads rather than triads at the AI junction. So much for cardiac muscle. The most important point is there's no function, for most of us, there's no functional regeneration of cardiac myocytes, which means that you need to really be careful with your health. Smooth muscle cells. This will be even less detailed. Comprised of these spindle-shaped or fusiform cells, organized into groups or sheets, and then again they're connected by gap junctions, so they tend to contract as a wave all at once. These are single cells, the nucleus is centrally located, and the organelles are juxtanuclear or polar in position on either side of the nucleus, in other words. And they are also surrounded by a basal lamina and an endomysium. That's not true for cardiac muscle. There might be an endomysium, but there's no perimysium or epimysium. There are contractile myofilaments. Actin and myosin are present in smooth muscle, but they're not in registration with each other, so you don't have a stride appearance to smooth muscle. There are two places that are referred to as anchor points, where you would find these actin and myosin filaments sort of anchored. Some of these occur at the edge of the cell membrane. They're called focal densities. There are additional proteins at that location. Typically, vinculin, tannin, and desmin are at the focal densities. There's a similar structure to a focal density, but found in the middle of the cytoplasm. They're just called dense bodies. There's a different set of proteins there, mainly alpha-actinin are in the dense bodies. Come on. Smooth muscle cells can maintain a contracted state for long periods of time, apparently without much fatigue, and that manner of contraction is called a latch state. Smooth muscle cells get sort of thicker when they contract. When needed, smooth muscle cells can increase show an increase in both size, it's called hypertrophy, and or they can increase their overall numbers, in which that would be hyperplasia. So you think of a woman about to give birth or undergoing gestational changes, the uterus gets much larger, the cells increase in number, more smooth muscle cells in the uterus, and they get longer too. And most interesting, in the last 5 or 10 years, this is mainly from people studying the gastrointestinal system, they've observed that even when smooth muscle cells are not involved in a reparative process, they may gradually increase their numbers, and that's been seen in the terminal parts of the GI system, like the large bowel. I don't think anybody knows exactly the significance of that, but it's been noted. Again, that sounds like a graduate student project. And we want you to cut 10 feet of large bowel in cross-section and count smooth muscle nuclei. Yeah, no thanks. Yeah. Do you have another project in mind? Yeah. Here's what you would see in a typical light microscopic section of smooth muscle. These spindly cells, fusiform, if you want to like that term. Nucleus is in the center, but remember when you get them in a cross-section at the edge here, you're not going to catch the nucleus in every one of these because some you're getting in the middle and some you're hitting the edges or ends of the cells. The organelles are described as being in a juxtanuclear position, which means they're on either side of the nucleus. They have the typical features of any cell, rough ER, smooth ER, mitochondria, Golgi. They do have Golgi and rough ER because in addition to contraction, smooth muscle cells produce extracellular matrix material. So they produce different collagens and different extracellular fibers. The thing that's outlined in the box is the cytoplasmic densities. A density, that's sort of an anchor point for actin and myosin within a cytoplasm. There's a similar thing, it's not shown very well in this image, that occurs under the plasma membrane of these smooth muscle cells. Those are the focal densities. You can see the focal densities better on this image. We've got maybe 8 smooth muscle cells, probably a cross section here. Only one has got a nucleus in it, so you can see the cytoplasmic densities here all the way through these guys, and then these focal densities underneath the plasma membrane, those are also anchor points for the actin and myosin, okay? This image shows you something else that I'm not sure you'd recognize it, but this thing labeled 5 here and 5 down here, those are autonomic nerve terminals, those are autonomic axon terminals, which is one of the ways that smooth muscle is caused to contract. There are some other ways too. And then there should be gap junctions between the sides of these smooth muscle cells because of other than their innervation by autonomic fibers, the wave of depolarization spreads via gap junctions. Smooth muscle cells are obviously capable of mitosis. They don't need stem cells. They're their own stem cell. And that's how they undergo replacement, by undergoing mitotic replacement. There are some ways that smooth muscle cells can differentiate from, stem cells in the adventitia of blood vessels. That's been documented. And it's also been shown that some come from the division of endothelial cells and pericytes after injury. That's a bit of a controversy, but some people believe that. If you think about it, when we talked about the skeletal muscle and cardiac muscle, we had something that held and stored ionized calcium, at least for a while. In smooth muscle, there is rough ER and smooth ER, but it doesn't function like it does in other muscle. In other words, it's dedicated for either making steroids or extracellular matrix material. So we need something that holds and stores the ionized calcium, and those are called caveolae. The term is caveolae. They look like pinocytotic vesicles if you see them, but they're called caveolae, I guess, because we're dealing with smooth muscle. They function to the T-tubule or transverse tubule system in that they control entry and storage of calcium until contraction is required for the smooth muscle cell. You still need ATP, and you still need the sliding movement of actin with respect to myosin to get shortening of smooth muscle cells. As I said, because these also produce extracellular matrix components, they have rough ER and smooth ER, so those can't be dedicated to storing calcium. So that's why we have the caveolae. And there are pictures in your textbook of how the caveolae look. They look like pinocytotic vesicles to everybody. In addition to electrical depolarization via the autonomic nervous system, other things that cause smooth muscle to contract are mechanical distortion, and biochemical modulators are substances. So that's different than other muscle cells. These other mechanisms that cause smooth muscle to contract are unique to smooth muscle: mechanical distortion and biochemical stimuli. Okay, I think that's my last frame. Yep. Let's take a break for a bit and then do whatever amount of laboratory work.